welcome to my channel Upma Biology classes. In this video, our topic is immunity. It means disease resistance. This video is taken from Chapter 8, Part 3 of Immunology series, which is the part of CBSE Class 12. So let's start the topic. At first, which is important for us to know that what is immunity. Daily we are exposed to large number of infectious agents. However, only a few of these exposures result in disease. The reason is that our body is able to defend itself from most of these foreign agents. This ability of the body to fight against the disease-causing organism conferred by the immune system is called immunity. So, the definition of immunity is the ability of an organism to recognize the foreign material that enter the body and to mobilize the cells and cell products to speedily remove that foreign material. The study of body's natural defense mechanism against pathogens and toxic molecules is known as immunology. And vulnerability of a person to infectious agents is called susceptibility, which is opposite of immunity. Now, the question is, how can we develop our immunity? So in development of immunity, there are three different ways in which the first is immunity through disease, second is vaccination, and the third is antitoxins. So these three are the development of immunity. So in which the first one, that is immunity through disease. Response of a person to the infection of a pathogenic organism varies through time. The first time he gets infection of a pathogen, like virus which is responsible for mumps, he is likely to develop the disease which is mumps. Subsequent infection of the same microorganism does not cause the disease. So let us find out the reason. In the case of first infection, antibody formation is slow and the virus has time to multiply and spread throughout the body. Thus, disease appears. Eventually, antibody formation picks up and the disease disappears. The cells capable of producing antibodies persist in the body as memory cells for a long time, maybe for life after first infection. They quickly become active on further infection and produce antibodies of the same type. The antibodies rapidly overcome the infection fully. Hence, the disease does not appear again. This is the basis of disease resistance or immunity. Now, here, what is the duration of immunity? So, some disease such as measles, mumps, smallpox, polio, once caught, produce a lifelong immunity for other disease such as diphtheria, typhoid fever. Immunity lasts for years but not for life. Still, other diseases such as common cold and flu generate only a short-term immunity. Viruses of cold and flu is spread from cell to cell and not through the blood. Therefore, antibodies of the body fluid cannot efficiently attack the pathogens. Moreover, influenza virus has a high mutation rate and new strains develop in a short time. Antibodies formed for one strain are not effective against another strain. Hence, every year, flu epidemic sweeps the world. Now, the second way of immunity development is vaccination. So, what is vaccination? Vaccination is a technique to develop immunity without infection. Here, weakened, that is attenuated or dead pathogens or portion of pathogens are injected into a person who is required to be made immune. The pathogens given in a vaccine are unable to cause the disease but are sufficient to stimulate the formation of antibodies by the host immune system that recognize the antigens. Thus, a vaccinated person develops immunity against the pathogen without contracting the disease. Today, vaccines are available against smallpox, cholera, measles, mumps, polio, and rabies. Often 2 or 3 additional doses are needed to generate adequate immunity. These are called booster doses. Smallpox was the first disease to be eliminated by vaccination. It is also the first disease to be officially declared wiped out by the human efforts. So this is all about vaccination. After this, the third way is antitoxins. So what are antitoxins? Antibodies that neutralize toxins produced in the body. Bacterial toxins are introduced from outside like the snack venom. So the toxins which produced in the body like the bacterial toxins and which are introduced from outside are the snack venom are called antitoxins. Antitoxins are these days prepared artificially and used as a remedy for the snack bite. So how can we produce antitoxins? Antigen, like the snack venom, is injected in low doses into an animal like horse, rabbit, etc. After the animal develops antibodies for the antigen, its blood is drawn and allowed to clot. The serum left behind contains antibodies. It is injected into a person bitten by a snake to neutralize the snack venom. This treatment is used in cases where the affected person is likely to die, as he would not be able to produce sufficient amount of antibodies quickly in his own body. It, however, gives temporary immunity against the antigen, only for a few hours or a few months. So this is all about the antitoxins. After this, now types of immunity. So we will discuss each and every part in detail here. That what is types of immunity. There are 2 types of immunity, in which the first one is here innate immunity and the second is acquired immunity. So in case of innate immunity or inborn immunity, certain animals do not suffer from some infectious disease. The reason is that the microbes that causes these diseases fail to flourish in such animals. This type of immunity is inherited by the organism from the parents and protect it from birth throughout life. It is known as innate or inborn immunity. Like human beings, example, have innate immunity against distemper or fatal disease of dogs. While in case of acquired or adaptive immunity, this is the immunity developed by an animal in response to a disease caused by infection of microbes. It is specific and is mediated by the antibodies or lymphoid cells or both, which make the antigens inactive or harmless. It not only relieves the victim of the infectious disease but also prevent its further attack in future. Now, acquired immunity is of 2 types, in which the first one is natural or active immunity and the second is artificial or passive immunity. So, active immunity is said to be active when an organism's own cells produce antibodies. It develops when a person suffers from a disease or gets vaccination for a disease. While in case of passive immunity, immunity is said to be passive when antibodies produced in other organisms are injected into a person who already has a potential antigen in his body. Passive immunity is developed to counteract snack venom, rabies, tetanus toxins and salmonella infection. So here you can see a difference between our immune system. In case of innate immunity, there are some cells like the macrophages, neutrophils, dendritic cells, eosinophils, basophils. While in case of adaptive immunity, antigen presenting cells like the professional APCs, non-professional APCs, T cells like the memory T cells, effector cells, cytotoxic cells, helper cells, regulatory cells, etc. And B cells like the memory B cells and the plasma B cell. So here 5 types of cells are present in innate immunity, while in case of adaptive immunity, there are three types of cells, that is antigen presenting cells, T cells and B cells. So, what are the difference between the innate immunity and the adaptive immunity? So here you can see that innate immunity shows the rapid response while the adaptive immunity shows the slow response. Innate immunity is a non-specific type of defense present since birth. It is accomplished by providing different types of barriers to the entry of the foreign agents into our body. These barriers are physical barriers, physiological barriers, cellular barriers, cytokine barriers, and all these barriers are discussed in detail later in this chapter. While in case of adaptive immunity, here the acquired or adaptive immunity lasts for the whole life of an animal in the case of certain disease and for a few years in case of other disease. Such disease has already been mentioned here. So, the memory cells formed by the B and T cells are the basis of acquired immunity. They have long lives and provide antibodies and killer T cells in subsequent infection. So, this is the main difference between the innate immunity and adaptive immunity. Now, here you can see that in case of innate and adaptive, physiochemical barriers are skin mucous membranes in innate immunity and cutaneous and mucosal immune systems, antibodies in mucosal secretions in adaptive immunity. According to the cells, phagocytes like the macrophages, neutrophils and natural killer cells are present in innate immunity and the lymphocytes are present in adaptive immunity. According to soluble mediators active on other cells, here macrophages, derived cytokines, example alpha and beta interferons, tumor necrosis factors, etc. are present in innate immunity, while in case of adaptive, lymphocyte derived cytokines and the example is interferon gamma. So this is the physiochemical barrier cells and the soluble mediators which is active on other cells in case of innate and adaptive immunity. So what are the main differences between the innate and acquired immunity? In case of innate, the defense mechanisms that are not antigen specific and immediately comes into play on antigens appearance in the body, therefore are also known as non-specific immunity, while acquired immunity has the defense mechanism that is not always present but involve antigen specific immune response, therefore also known as specific immunity. In case of innate immunity, it is present since birth, hence also known as natural immunity, while the acquired immunity is acquired during the lifetime, thereby also known as acquired immunity. Innate immunity, it's the first line of defense of immune system, while the acquired immunity is the second line of defense, the action against pathogens that are able to invade or overcome. In case of innate immunity, response time occurs rapidly from minutes to hours, while acquired immunity response times occurs over days to weeks. So these are the main difference between the innate and acquired immunity. So here you know that acquired immunity is of two types, in which the first one, that is the active immunity, develops in response to an infection or vaccination, which is of two types, natural and artificial. Natural are like the antibodies developed in response to an infection, and the artificial is antibodies developed in response to a vaccination. While in case of passive immunity, develops after you receive antibodies from someone or somewhere else. And it is also of two types, natural and artificial. Natural like the antibodies received from mother, example through breast milk. And the artificial are the antibodies received from a medicine, and the example from a gamma globulin injection or infusion. So this is all about the active and passive immunity. So in case of active immunity, infection of pathogen or injection of vaccine, then the immune response occur, then the antibody produced and the memory cells retained, then infection destroyed, then infection by the same pathogen, then new rapid immune response occur. So this is the way of active immunity. While in case of passive immunity, antibodies acquired from a host mother via placenta or milk, that is colostrum. Immunized organism, example horse or monoclonal antibodies. Then infection destroyed, then no memory cells created because the body has not produced an immune response itself. So further infection possible by the same pathogen. So in case of active immunity, rapid immune response occur, while in case of passive immunity, further infection possible by the same pathogen. Now, what is infant's immunity? So, there is another form of passive immunity, which is a newborn baby is passively immune, being temporarily protected from disease by the antibodies that is received from the mother's blood before birth. Nursing mothers also transfer antibodies prepared in their body to the infants in their milk. After a few weeks, infant's own immune system start working and the mother's antibodies are no longer needed. Bottle-fed infants do not get this benefit. So this is all about the infant immunity. After this, what is the difference between the passive and active immunity? Passive immunity, here the no active host participation, that means received passively, while in case of active immunity, produced actively by the host immune system. In case of passive immunity, antibodies transfer directly, while in case of active immunity, antibodies induced by infection or by immunogens. In case of passive immunity, is due to ready-made antibodies, while in case of active immunity often involves both the cell-mediated and humoral immunity. In case of passive immunity, types like the natural, where the transfer of maternal antibodies through placenta, and the artificial, that is injection of immunoglobulins. While in case of active immunity, types like the natural, that is clinical or inapparent infection, and the artificial, that is induced by vaccines. Passive immunity is immediate immunity, no lag period, while the active immunity, here the immunity effective only after lag period. Passive immunity is transient, less effective, while the active immunity is durable, effective protection. In case of passive immunity, no immunological memory, while in case of active immunity, immunological memory present. In passive immunity, subsequent dose less effective due to immune elimination, while in active immunity, booster effect on subsequent dose. In case of passive immunity, no negative phase, while the negative phase may occur in active immunity. Passive immunity is applicable even in immunodeficient, while the active immunity is not applicable in the immunodeficient. So these are the main difference between the passive and active immunity. After this, what is the history of vaccine and vaccination? So, idea of vaccination was conceived by Edward Jenner, a country doctor during a dreadful smallpox epidemic in England. He found that epidemic killed large number of people in cities but seldom affected rural people who walked around cattle. So, most of the farmers and dairymen had suffered from cowpox and had recovered. This led him to think that an attack of cowpox had made these people immune to smallpox and that vaccinating people with smallpox could protect from smallpox. So, the first vaccination here, the Dr. Jenner tested his vaccination theory in May 1796 on James Phillips, a healthy boy of about 8 years. It is really credible for James' mother to offer her young son for the test. Dr. Jenner took James to a dairymaid, Sarah Nelms, whose hand had developed a cowpox pustule following an infection from her master's cow. Dr. Jenner made two shallow cuts on James' arms and inoculated them with the matter taken from Sarah's cowpox pustules. James' arm developed a pustule which forms a scab and healed, leaving only a scar. Did this make James immune to smallpox? The only way to ascertain this was to inoculate him with the smallpox. Now, in case of smallpox inoculation, in July of the same year, Dr. Jenner inoculated James with material from a smallpox pustule. No sign of smallpox appeared. Inoculation was repeated after several months. Again, smallpox did not develop. Dr. Jenner's vaccine was successful. So, James Phillips had certainly become immune to smallpox. The antigen of smallpox and cowpox are so similar that the same antibodies work against both of them. So, according to this, Jenner's conclusion is Jenner proposed that inducing the mild form of a disease stimulates the body to develop an active immunity for protection against the virulent form. He used the term vaccine for the immunity producing preparation and the term vaccination for the process of inoculating the preparation into the body. The use of a vaccine for the treatment of a disease is called vaccinotherapy. So, an experiment is designed to test the hypothesis is Jenner injected a boy with a cowpox and later injected him with a smallpox. So, interpretations of result is Jenner tested the boy for smallpox and found that he did not have it. So, the conclusion according to this experiment is Jenner concluded that a person who once had cowpox would not get smallpox. After this, Pasteur confirmed Jenner's findings and produced vaccines for anthrax, chickenpox, cholera, and rabies. Now, vaccines are also available for diphtheria, cholera, typhoid, whooping cough, tetanus, tuberculosis, plague, measles, mumps, and polio, etc. The cells infected with the cancer-causing viruses usually show on their surface two new antigens, one of which is specific to the virus. This has led to the first successful immunization against cancer in cats and chickens. Unfortunately, efforts to produce antibodies to cancers not caused by viruses have had very little success. Many serious infections diseases also have no effective vaccines. These include malaria, trypanosomiasis, and AIDS. The pathogens of this disease frequently change their surface antigen. So this is the history of vaccine and vaccination. After this, now the defense mechanism of body against pathogens. So the organism have evolved a variety of defense mechanisms against the disease-causing viruses and organisms. These mechanisms maintain homeostasis to keep the organism alive. After death, the organism lose their defense mechanism and are easily attacked and rapidly destroyed by microorganisms. So, what is the immune system? The immune system is a complex network of cells and proteins that defends the body against infection. The immune system keeps the record of every germ it has ever defeated so it can recognize and destroy the microbe quickly if it enters the body again. The study of body's defense mechanism against pathogens is called immunology. Now, kinds of defense mechanism. So, there are two kinds of defense mechanism against microbes in which the first one is non-specific defense mechanism and the second is a specific defense mechanism. So, non-specific defense mechanism is similar for most type of infection. Hence, its name is non-specific. It resists infection by blocking the entry of pathogens into the body or by destroying the microbes through means other than antibodies. So the non-specific defense mechanism is of two types, but the specific defense mechanism provides protection against specific foreign materials and is often called the immune system. This system forms the third line of body's defense against microbes and harmful molecules, while the non-specific defense mechanism so first line of defense and also second line of defense. Now, non-specific defense mechanism is of two types, in which the first one is external defense, where the defense comprises physical and physiological barriers to the entry of the pathogens into the body, and the second is internal defense. So, in case of this, it includes cellular barriers and the cytokine barriers. Now, in external defense, here it is of many types, like physical barriers and physiological barriers, while the internal defense is of only two types. First one is cellular barriers and the second is cytokines barriers. So we will discuss each and every part in detail in the next slide. So here you can see all the first line, it means the external defense mechanism. So first one, that is the physical barrier here. This in turn are two types. First one is the skin and the second is mucous membrane. So in the first picture you can see the skin. The skin provides a nice protective covering to the body. Its outer tough layer called horny layer or stratum corneum consists of a dead fully keratinized cells. These cells contain a hard insoluble fibrous protein called keratin or horn instead of soft protoplasm. The horny layer is waterproof and germ-proof. It successfully prevents the entry of viruses and bacteria. Now, mucous membranes, which is also a physical barrier. The digestive, respiratory and urinogenital tract open out at one or both the ends and do not have a direct communication with other parts of the body. The parasites present in these tracts are not in the physiological interior of the body. The mucous membranes lining these tracts are therefore treated as a part of the external defense like the skin. Now, in case of mucous membrane, there are many organs like the buccopharyngeal cavity. Dust particles and microbes entering the buccopharyngeal cavity via mouth are caught in the mucus and eliminated with sputum. A coating of mucus over the intestinal lining also traps the microbes for removal in the feces. Next is respiratory tract. Microorganisms and dust particles often enter the respiratory tract with air during breathing. Many of these are caught in the mesh of hair present in the nostril. Those which pass through this filtering device are trapped in the mucus that covers the respiratory tract. The cilia sweep the mucus loaded with the pathogens and dust particles into the pharynx. From here, it is thrown out or swallowed for elimination with the feces. After this, now eyes, which is also has a mucous membrane. Secretion of tears and the movements of eyelids flush out the microorganism settling on the eyeballs from the air. Now internal tracts. The various tracts in the body are flushed with fluid such as saliva, digestive juice, bile and urine so that the microbes tends to swept away. After this, the physiological barrier, in which first one, that is the skin secretion and bacteria. The oil and sweat secreted by the sebaceous and sudoriferous glands contain fatty acids and the lactic acid, which make the skin surface acidic, that is pH 3 to 5. This does not allow the microorganism to establish on the skin. The skin harbors some friendly bacteria which release acids and other metabolic wastes that check the growth of microbes. Now, the second physiological barrier is saliva. Saliva also contains lysozymes which kills the microorganisms that are not the normal inhabitants of the buccal cavity and come with food and drinks. Now, gut secretion and bacteria. The microorganism may escape the action of saliva and reach the stomach with food and drinks. Bacteria trapped in mucus in the respiratory tract also reach the stomach when swallowed. Here they are killed by hydrochloric acid and proteolytic enzymes of the gastric juice. The proteolytic enzymes in the small intestine also help in killing the microorganism. If the bacteria survive and reach the large intestine, they are attacked to the gut microorganism which secrete antibiotics that kill many pathogenic bacteria. Now, next is bile. Bile is a bitter alkaline pH which is 8 secretion of the liver checks the growth of foreign bacteria on the semi-digested food, that is the chyme in the intestine. Now tears. Tears, a slightly saline fluid secreted by the lacrimal glands over the eyes, also contains lysozyme which prevents eye infection. Next is nasal secretion. Nasal secretion also destroy the harmful foreign germs with their lysozyme. Now cerumen. Cerumen, which is the earwax, a bitter brownish secretion from the ceruminous gland into the auditory canal, traps dust and bacteria. It contains an effective antibacterial component. It also prevents entry of insects. Next is vaginal bacteria. Certain bacteria like lactobacilli normally live in the vagina. They produce lactic acid from glycogen of the cells that periodically break off from the mucous membrane. Lactic acid kills the foreign bacteria. The lactic acid bacteria of the vagina form female's best natural defense against infection. Hence, frequent vaginal douches. So, douches means wash with a jet of water, particularly with disinfectants, should be avoided. Douches remove the friendly bacteria. Now, penetration of the first line of defense. So here you can see the first line of defense like the saliva, skin, tears, mucus, good gut bacteria, stomach acid, etc. Skin and mucous membranes may fail to keep out the invaders. Some parasites make way through the skin that like hookworm. Others enter through wounds and opening of sweat glands and hair follicles. Still others are injected by blood-sucking arthropods. So, the microorganism may injure and pass through the thin, moist, relatively vulnerable mucous membranes of digestive, respiratory, and urinogenital tracts and get into the tissues or blood. It is therefore very essential for survival to have a second line of defense for controlling the parasites that have entered the body by breaking through the first line. After this, now the second line of defense, which are cellular barriers and cytokine barriers. So, in case of cellular barriers, the body's internal defense is carried on by the cellular barriers which include white blood corpuscles, macrophages, inflammatory reactions, fever and complement system. All these devices operate together to check damage to the body by the pathogens. Often the invaders are overcome and the host recovers. Now, in case of cellular barrier, first is white blood corpuscles, that is leukocytes. Number of leukocytes increase in response to infection. Rise in WBC count is called leukocytosis. Here you can see the leukocyte moving out through ciliary wall into the tissue spaces. So, the leukocytes creep out of the capillaries by the amoeboid movement into the intercellular spaces. If there is an infection, this process is called diapedesis. The leukocytes are the key to our survival in the daily war against infection. They help in different ways, in which the first way is here you can see, which is the migration movement of the leukocyte, that is the diapedesis. Here you can see that is neutrophil, then the rolling, then capture, then adhesion and activation, then spreading, then extravasation, then phagocytosis of the bacteria. So this is the migration of white blood cells. Now, in the white blood corpuscles, first is the neutrophils. These cells seize, engulf and digest the microorganism infecting the body tissues, and they are called phagocytes, and the process is known as phagocytosis. If an invader enters the bloodstream, the chance of a phagocyte capturing it is remote in the churning blood. It is then disposed of by other phagocytic cells are called macrophages referred to ahead. Next is monocytes. They are motile and occasionally show phagocytic activity. Generally, they change into macrophages. Now, in case of macrophages, in the picture you can see the structure of macrophages. These are large phagocytic cells that arise by enlargement of monocytes. They are of two types, fixed and wandering. So, the wandering macrophages occur in the connective tissues throughout the body. They move to the site of infection like the leukocytes and disposed of the microbes. They are called histocytes also. Now, the fixed macrophages are located in the lining of the blood and lymphatic capillaries in spleen, lymphoid nodes, liver and bone marrow. They catch hold of the microbes and dead cells that are carried along in the blood and lymph. The wandering and fixed macrophages together form the reticuloendothelial system. Many macrophages may fuse to form a large multinucleate giant cells to capture a foreign body too big to be enclosed within a single macrophage. Now, soldiers and scavengers. So the phagocytic cells, that is neutrophils and macrophages, besides destroying the microbes, also eat up the dead tissue cells. They are popularly called the soldiers and scavengers of the body. Now, if we talk about the survival within the macrophages, some microbes such as here Mycobacterium tuberculosis, then Shigella, then Leishmania, then Salmonella manage to survive within the macrophage with special adaptations. So this is all about here the barriers, that means white blood cells, in which the first one is the neutrophils, then monocyte, then macrophages. Now here the next is inflammatory reactions. So what is inflammatory reactions? Injury like the bruise, cut, burn or bite of an animal allows the foreign matter to enter the tissues. What are the foreign matters? Like the bacteria, viruses, fungal spores, etc. Certain substances released by the damaged cells initiate the formation of blood clot. The clotting process checks the loss of blood from the injury. Foreign matter in the wound produces toxic substances which kill mast cells. These broken mast cells release histamine. Histamine causes dilation of capillaries and small blood vessels surrounding the injury and increase the permeability of the capillary wall. As a result, more blood flows to that area, making it red and warm, and fluid leaks out into the tissue spaces, causing its swelling. This reaction of the body is called inflammatory response and is a part of the internal defense. So in the process of here, that means inflammatory reaction, here the process, that is first the bacteria and other pathogen enter wound. Platelets from blood release blood clotting protein at wound site. Then mast cells secrete factors that mediate vasodilation and vascular constriction. Delivery of blood plasma and cells to the injured area increases. Then neutrophils secrete factors that kill and degrade pathogens. Then neutrophils and macrophages remove pathogens by phagocytosis. Then macrophages secrete hormones called cytokines that attract immune system cells to the site and activate cells involved in tissue repair. Then the inflammatory response continues until the foreign material is eliminated and the wound is repaired. So this is all about the events which occur in the inflammatory reaction. So the major events in the local inflammatory responses are: first, the chemical signals released by the activated macrophages and the mast cells at the injury site causes nearby capillaries to widen and become more permeable. Then fluid, antimicrobial proteins and the clotting elements move from the blood to the site clotting begins. Then chemokines released by various kinds of cells attract more phagocytic cells from the blood to the injury site. And the last is neutrophils and macrophages phagocytose pathogens and cells divides at the site and the tissues heals. So this is the major events in the local inflammatory response. So, in case of tissue injured caused by physical and chemical agents or pathogenic microorganism, first one, that is the capillary widening, increased blood flow, that is heat created. Then increased permeability, that is fluid release into tissue, then redness cause, swelling cause. Then attraction of the leukocyte, then extravasation of the leukocyte to the site of injury, then tenderness occur. And the last is systemic response, that is fever and proliferation of the leukocyte, that is pain occur. So this is the way of the tissue injury caused by the physical and chemical agents or pathogenic microorganism. Now, what is pus? Many neutrophils and the macrophages are killed by toxins released by the invaders. The zone of dead phagocytes is enclosed by living phagocytes and the microbes are trapped inside. So, the barrier usually prevents further spreading of the infection. The microbes are gradually disposed of by the phagocytes. The dead phagocytes and the damaged cells leave the body in the form of the pus, and the formation of pus is a sure sign of infection. Now, what is fever and how can it occur? The inflammatory response may be localized or systemic. The localized response is confined to the site of injury only. The systemic response affects the entire body. In this case, the WBC count of the blood increases, body temperature rises and causing fever. So, this may be brought about by a protein called endogenous pyrogen, which is released by macrophages. When enough pyrogen reaches the brain, the body's thermostat is reset to a higher temperature, allowing the temperature of the entire body to rise. The endotoxins of gram-negative bacteria also cause fever. Mild fever strengthens the defense mechanism by activating the phagocytes and by inhibiting the growth of microbes. Higher body temperature reduces blood iron level. So, bacteria need more iron as the temperature rises. Therefore, the bacterial growth is reduced in a fever-ridden body. A fever-reducing medication may counteract body's natural defense. A very high temperature may prove dangerous. It must be quickly brought down by giving antipyretics and by applying cold packs. Now, what is the complement system? This system assists or complements other defense mechanisms of the body. So, it consists of about 20 proteins present in the serum. They are synthesized mainly in the liver. The complement proteins function in different ways. Some complement proteins rupture the cell membranes of the microbes. Others make mast cells to release histamine and strengthen inflammatory reaction. Still others attract the phagocytes to the infected area. So, this is the process of complement system. Now, what are the cytokine barriers? So, a special defense system works especially against viral infection. It has no effect on microorganisms. Cells invaded by a virus produce an antiviral protein called interferon. The latter is released from the infected and dying host cells. On reaching the nearby uninfected cells, it makes them resistant to the virus infection. Resistance is provided by the formation of certain proteins which prevent multiplication of the virus. Fever increases the production of interferons. Interferons have proved effective in treating influenza and hepatitis but have not succeeded against cancer. So, now organisms such as bacteria, rickettsias and protozoans have also been found to elicit interferon response from the cell. Now, the specific defense mechanism. So, what is a specific defense mechanism? This mechanism provides protection against specific foreign materials and is often called the immune system. This system forms the third line of body's defense against microbes and harmful molecules. So here you can see a structure of the antibody. The most important characteristic of the immune system is its ability to recognize body's own cell and macromolecule self from those which are foreign invaders, which are non-self. It tolerates the self but destroys the non-self. So the foreign matter that enters the body is called antigen, like the picture you can see, or immunogen. It stimulates the immune system to produce protective chemicals or special cells to destroy the antigens. The protective chemicals are called antibodies. So, the antigens entering a body may be virus, bacteria, or toxins such as snake poison. Usually, the protein molecules present on the surface of foreign material act as antigen, but some carbohydrate and lipid molecule can also act as antigens. Antibodies are proteins in nature. Each antigen causes the formation of a specific antibody. Antigens and antibodies have complementary reactive sites that fit together like lock and key. So the antibodies react with the antigens and make them inactive or harmless. So in this picture you can see the antigen antibody interactions like lock and key. Antibodies are globular proteins each with two identical reaction sites. Antigens are large protein or carbohydrate molecules, each with many different types of reaction sites, and antigen molecules combine with several antibody molecules. So, this is the main difference between the antigen and antibody. Now, here you can see the antigen and antibody reaction. That means how antigen binds to the antibody and show lock and key hypothesis. Now, here one thing you have to know that antigen and antibody interactions are of many types. Like here the first one, that is neutralization. Some antibodies neutralize the antigens termed toxins and make them ineffective. They are called antitoxins. The phagocytes dispose of the neutralized antigen antibody complexes. Next is agglutination, that is particulate antigen. Certain antibodies causes the particulate antigens like the bacteria red corpuscles stick together in clumps, thus immobilizing them for easy disposal by the phagocytes through ingestion. They are termed agglutinins. Next is precipitation, that is soluble antigen. Other antibodies combine with the antigens to form precipitate that are easily ingested by the phagocytes. They are known as precipitins. Next is here complement system. So, antibody antigen complexes activate complement proteins which may lyse cell walls of the bacteria causing their disintegration, incite inflammatory response, apponize antigens and attract phagocytes to area of infection. So, after this, one extra, that means fifth, antibodies fight with the antigens are the disposal of the antigens, which is of the fifth, that is opsonization. Some antibodies coat the surface of the microbes and make them more susceptible to phagocytosis. Such antibodies are known as opsonins. So these are the way of the antigen antibody interactions, which are neutralization, agglutination, precipitation, opsonization and complement activation. So in case of here you can see that in case of the whole process of the antigen antibody interaction, first one, that is the neutralization of microbes and toxins, then opsonization and phagocytosis of microbes, then antibody dependent cellular cytotoxicity, then phagocytosis of the microbes opsonized with complement fragments, then inflammation and the lysis of the microbes and complement activation. So these are the way of antigen antibody interaction. Now, structure of antibodies. Different types of antibodies can be produced in our body such as IgA, IgM, IgE, IgG and IgD. Each antibody has 4 peptide chains, 2 small or light chains and the 2 large or heavy chains. The antibody therefore is represented as H2L2. Now, the types and characteristics of antibodies and the distribution in the body. In this picture you can see the distribution. Now, IgG is the highest opsonization and neutralization activities classified into four subclasses IgG1, G2, G3 and G4. IgM produced first upon antigen invasion increases transiently and IgA is expressed in mucosal tissues, forms dimers after secretion. IgD unknown functions and IgG has involved in allergy. Now, this diagram showing an antigen with the epitopes. So, forms of antigens, if we talk about this, the microorganisms are not the only antigens that enter the body. Many other foreign materials also act as antigens to cause immune response. This includes pollen, egg white, feathers, some fruits and vegetables, chicken, blood cells from other persons, transplanted tissues and organs and medicines. Now, cells of immune system in which the first cell here, that is the lymphocyte. So lymphocytes are the main cells of the immune system. They, like the other blood corpuscles, arise from the stem cells, the hematocytoblast, present in the liver in the fetus and in the bone marrow in the adult. Some of them undergo differentiation in the gut-associated bursal lymphoid tissues and are called bursal or B lymphocytes. Others are differentiated in the thymus glands and are termed as thymus or T lymphocytes. So, the mature lymphocytes pass into the body fluid and circulate in the body. Many of them stay in the lymph nodes. The B lymphocytes and the T lymphocytes form humoral or antibody mediated immune systems and cell mediated immune system respectively. Both the immune system need antigens to come into action, but they respond in different ways. So, a common lymphoid progenitor, and here which is the stem cells in the bone marrow. After the processing here in the thymus, formation of lymphoblast, that is the T cells, and in case of the thymus, formation of the T cells. Now the first one, that is antibody mediated immune system, AMIS, or humoral immunity, which is caused by B lymphocytes. So, the M's defend the body against some viruses, bacteria with polysaccharide capsules, and the toxins that enter the body fluid, that is blood and lymph. Now, the question is how B cell works. So, in case of pathogens, B cells are of the two types, that is plasma B cells and the memory B cells. So, when antibodies on a B cell surface bind antigen, the B cell is activated and dividing, producing a clone of the daughter B cells. The daughter cells specialize into plasma B cells and memory B cells. So, B cells each recognize a different antigen. The correct one develops into plasma cells, clone of the correct B cells, which produces antibodies. So, the first meeting a pathogen, this process takes 10 to 14 days, and memory B cells subsequent meeting takes about 5 days. So this is how B cell works. Now, what is the way of formation of B cell? A B cell is triggered when its encounter its matching antigen. So the B cell engulfing the antigen and digest it. Then it displays antigen fragments bound to its unique MHC molecule. This combination of antigen and MHC attracts the help of a mature matching T cells. Cytokines secreted by the T cell help the B cells to multiply and mature into antibody producing plasma cells, and the last is released into the blood. Antibodies lock onto matching antigens. The antigen antibody complexes are then cleared by the complement cascade or by the liver and spleen. So this is the way of doing or work of a B cell. So in case of B cell, the plasma B cells are antibody factory. The antibodies pass into the circulate in the lymph to dispose of the antigens. For this, the antibodies selectively bind to the antigen, forming antigen-antibody complexes to destroy the antigens. While the memory B cells live for a long time and serve to quick dispose of the antigens in case reinfection of the same virus or bacterium occurs. Now, the binding to antigen. Here you can see the process. First one, that is binding to antigen on the B cell receptor. Then the chemical signal occur. Then it becomes plasma cell and releases antibodies. So this is the way of B cell works. Now here you can see the mature B cell. After the blasting of B cell, it forms the plasma cell and the memory cells. So in case of antibody mediated immunity, body contains millions of B cells which is located in the lymphoid tissue. Each responds to a specific antigen. Then it becomes activated in presence of foreign antigen, unprocessed antigen weak response, and need to process antigen for a stronger response. So, if we talk about the antibody action, the antibodies bind to their specific antigens and inactivate the invading microorganism or the foreign molecules so that these are conveniently disposed of by the phagocytes. So here you can see the antibodies attached to the B cell, then the antibodies in the blood secreted by B cell, and attachment of this, then the antibody mediated immune system occur. After this, here, difference between the B lymphocyte and the T lymphocytes. So there are some characteristics and the differences. First one, that is the ancestral origin. B lymphocytes origin is bone marrow and the T lymphocyte origin is also bone marrow. The site of maturational processing in case of B lymphocytes in bone marrow, while in case of T lymphocytes in thymus. In case of receptors for antigen, B cells receptors are antibodies inserted in the plasma membrane, highly specific. While in case of T lymphocytes, T cell receptors present in the plasma membrane are not the same as antibodies, highly specific. B lymphocytes bind with extracellular antigens such as bacteria, free viruses, and other circulating for foreign materials, while the T lymphocytes bind with foreign antigens in association with the self-antigens such as virus-infected cells. In case of B lymphocyte, the types of active cell is plasma cell, while in case of T lymphocyte, the type of active cell is cytotoxic T cells, helper T cells, regulatory T cells. In B lymphocytes, the formation of memory cell occur. In case of T lymphocytes also occur. In B lymphocytes, the type of immunity is antibody mediated immunity, while the T lymphocyte type of immunity is cell mediated immunity. In B lymphocytes, the secretory product is antibodies, while in the T lymphocytes, secretory products are cytokines. According to the function, B lymphocytes help eliminate free foreign invaders by enhancing innate immunity responses against them, provide immunity against most bacteria and a few viruses. While the T lymphocyte lies virus-infected cells and cancer cells, provide immunity against most viruses and a few bacteria, aid B cells in antibody production, modulate immune response. According to the lifespan, B lymphocyte life is short while the T lymphocyte life is long. So, according to the name, B cell has known as B lymphocyte, T cell known as T lymphocyte, origin in bone marrow and T cells in thymus. Position of the B lymphocyte outside lymph node and the position of T cells is interior of the lymph node. According to the receptor here, BCR, that is the immunoglobulin for the antigen, and TCR for the antigens. According to the connection, B cells can connect to the antigen right on the surface of the invading viruses or bacteria, while the T cells can only connect to virus antigens on the outside of the infected cells. Now, the difference between here the humoral and cell-mediated immunity. So, in case of humoral immunity, main cell involved is B lymphocyte, while in case of cell-mediated immunity, T lymphocytes involved. Where do cells develop? In case of humoral immunity, produced and mature in the bone marrow, while in case of cell-mediated immunity, produced in the bone marrow and mature in the thymus gland. What is antibodies? In case of humoral immunity, involve production of antibodies, while in case of cell-mediated immunity, does not involve production of antibodies. According to how are pathogens identified? In case of humoral immunity, via antigens floating in the blood, while in case of cell-mediated immunity, via antigens on the surface of the infected cells. How are pathogens killed? In case of humoral immunity, by antibodies, and in cell-mediated immunity, by specialized killer T cells. How do cells divide once they are stimulated? In case of humoral immunity, plasma cells divide into either plasma cells or memory cells, while in case of cell-mediated immunity, cells divide into different types of specialized T cells. So these are the main differences between the humoral and cell-mediated immunity. So, if we talk about here the cell-mediated immune response. So, at first, what is the cell-mediated immune response? The CMIS, that is cell-mediated immune response system, defends the body against viruses, fungi and some bacteria which have entered the host cells. Its defense cells cannot deal with the free antigen present in the body fluid. It also reacts against foreign tissues, transplants, and perhaps also against the body's own cells which become cancerous. The cellular immune response is given by T cells. These cells acquire the ability to recognize particular non-self cell surface and molecules in the thymus. On stimulation by contact with antigens, the T lymphocytes produced by division a clone of T cells, the lymphoblast. There are separate T cells for each type of antigen that invades the body. The T cells leave the lymphoid tissues and migrate to the site of infection. The T cells have a lifespan of 4 to 5 years or even longer. The T cells comprising the clone are committed T cells having a specific function. They are similar morphologically but of 4 types functionally: killer or cytotoxic cells, helper T cells, suppressor T cells, and memory T cells. Now, how does it work? T cells are mobilized when they encounter a cell such as a dendritic cell or B cell that has digested an antigen and is displayed antigen fragments bound to its MHC molecule. Cytokines help the T cell mature. The MHC antigen complex activates the T cells receptor and the T cell secretes cytokines. Then, some cytokines spur the growth of more T cells, and some T cells become cytotoxic cells and track down cells infected with the viruses. Some T cells become helper cells and secrete some cytokines that attract fresh macrophages, neutrophils, other lymphocytes and other cytokines to direct the recruit once they arrive on the scene. So, here according to the types, first one, that is the killer T cells. These cells migrate to the site of infection. They have surface peptides that act as receptors for the foreign antigens. When a T cell encounters a non-self cell, its surface receptor draws the two cells into a physical contact. The killer T cells here, they secrete a protein porphyrin which punctures the invader cell's membrane, and this kills the non-self cell. While the helper T cells, these cells stimulate the B cells to produce antibodies, and the suppressor T cells, which inhibit the immune response of both T and B lymphocytes to foreign antigens when infection is controlled. Now, memory T cells. These cells keep ready to mount a rapid and vigorous attack as soon as the same pathogen infects the body again. The killer T cells are the effector cells of the CMIS, whereas the helper T cells and the suppressor T cells are the regulator cells of the both AMIS and CMIS. One thing, CMIS is cell-mediated immune system and AMIS means antibody-mediated immune system. The cell of the AMIS is B lymphocytes and the cell of CMIS is T lymphocytes. Since the T cell must be present on the spot to play their role, they are said to form cell-mediated immune system. After this, now, what are the role of macrophages? Before this, difference between the interferons and antibodies. So what are the main differences? Interferons are produced by any micro-infected cells, while the antibodies are produced by plasma B cells only. Interferons leave the infected cell and enter a nearby healthy cell to dispose of the microbes, while the antibodies pass into and circulate into blood and lymph to dispose of antigens. Interferons induce the healthy cell to synthesize antimicrobial proteins that check microbial multiplication, while the antibody selectively binds to antigens that are immobilized for easy attack by phagocytes. Interferons are quick in action but gives a temporary protection against microbes, while the antibodies are slow in action but gives a long-lasting protection against pathogens. Interferons act inside the cells while the antibodies act outside the cells. Interferons form the body's second line of defense while the antibodies form the body's third line of defense. So these are the main difference between the interferons and antibodies. After this, now the role of macrophages in immune system. So the macrophages are among the first cell that respond to the infection. So they engulf the microbes and display the latter's antigen to alert the lymphocytes. The antigen is held onto the surface of the macrophages by a protein which marks the macrophages as the self, that is, as body's own part. All body cells carry this protein on the surface. With the flag of antigen, the macrophages move to the nearest lymph node. So this is the way. After this, that means the helper factors. Here the helper factor is stimulate here the two types of cells. First one is the killer T cells and the second is B cells. Killer T cells secrete perforin and the B cells secrete or produce antibodies. Then both combine to show the effect, that means destroy the antigens or microbes. So here it meets the various kinds of lymphocytes. A helper T cell recognize the macrophages antigen flag and join it. This start immune reaction. At the same time, the macrophage secretes endogenous pyrogen which activate and stimulate the T cells and B cells to grow and proliferate and causes fever to slow down microbial activity. Thus, macrophages, B cells and the T cells interact to produce immune response. So this is all about the role of macrophages in the immune system. After this, now what are the memory cells or the primed cells? So, after the infection disappears as a result of antigen-antibody interaction in the killer T cells, non-self cells interaction, some of the specific lymphocytes remain in the lymphatic tissues as memory or primed cells, ready to produce the antibodies and killer cells if the same antigen reappear. This is why the second attack of the infectious disease elicits quick and abundant antibody formation. The memory cells can give rise to more effector cells and the memory cells in case of a second attack of antigens, whereas the effector cells have a life of a few days only. The memory cells live long and some even for the whole life. This is why some diseases that attack in childhood do not attack again. The memory cells are stored in the spleen and lymph nodes. Now, what is the difference between the antigen versus antibody? An antigen is any substance that triggers an immune response in the body, while the antibody is a blood protein that is produced against a specific antigen. Antigen also called immunogens, while the antibody is also called immunoglobulins. Antigens can be either proteins, carbohydrates, lipids or nucleic acids, while the antibodies are the glycoproteins. Antigens interacting domains with the antibody is called the epitope, while the antibody variable site can bind to the epitope. Antigen causes either disease or allergic reactions, while the antibody protects the body from antigens either by immobilizing the antigens or lysing the pathogens. Antigens, the four types including exogenous antigens, endogenous antigens, autoantigens and neoantigens, while the antibodies are also of the four main types, that is IgM, IgG, IgE and IgA. IgD functions recently unknown. Now, the difference between the immunoglobulins versus antibodies. So, immunoglobulin refers to any class of structurally related proteins in the serum and the cells of the immune system which functions as antibodies, while the antibody refers to a globin protein which is produced by B cells in response to a particular antigen. Immunoglobulin occurs on the surface of B cell, while the antibody freely occurs in the circulation. Immunoglobulins has a transmembrane domain in order to attach to the plasma membrane of the B cell, while the antibody does not have transmembrane domain. Immunoglobulin 5 classes are IgG, IgM, IgA, IgD and IgE, while the antibody are particularly antibody specific to a particular pathogen. Immunoglobulin functions depends on the type of heavy chain, while the antibody non-self antigens are recognized by specific antigens and are neutralized by antibodies. So these are the main difference between the immunoglobulins and antibodies. Now, what are the main difference between all the types of antibody? So, IgG, which is the most common antibody circulating in the blood, facilitating phagocytic destruction of the macroorganism. IgG are the monomers. Subclasses are 4, which is IgG1, G2, G3 and G4, have the highest opsonization and neutralization activities, and the serum concentration is 75%. While in case of IgM, immunoglobulin of higher molecular weight including primary antibodies released into the bloodstream early in the immune response. Pentamer, there are no subclasses. The first antibody transiently increased upon antigen invasion. Serum concentration is 10%. In IgA, immunoglobulins playing a crucial role in the immune function of the mucous membrane. Monomer are dimer. Subclasses are 2, IgA1 and A2. Expressed in mucosal tissue, preventing colonization, serum concentration is 15%. In IgE, immunoglobulins produced in response to an allergy. Structure is monomer, involved in allergy, and the serum concentration is less than 0.01%. Here IgD, immunoglobulin, which is expressed in the plasma membranes of immature B lymphocytes. Structure is monomer, function as an antigen receptors on the activated B cells, and the serum concentration is less than 0.5%. So these are the main difference between all the types of antibodies, that is immunoglobulins. Now, before this is the end of the video in which we know about all the type of immunity, what is immunity, defense mechanism, vaccine, vaccination and non-defense mechanism, defense mechanism, role of CMIS, role of AMIS and memory cells, prime cells and all the differences. So this is the end of video. After this, the next upcoming video will be on the disorders of the immune system. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries, any suggestions or any comment, you can ask in the comment section below. Thank you for the watching.